Hi, everyone, and thank you for joining us, excuse me, for our live virtual MDA Engaged Community Education Seminar, where we are highlighting specialists in the Southeast today. I am Nicole Petrowski. I am the Community Education Specialist at MDA, and we appreciate you joining us this morning, and we're glad to be able to share this important information with you. We're committed to community education, and we believe in the power of bringing community together for opportunities to learn from specialists and having opportunities to connect with others. So thank you for being with us today. This is our final community education seminar for 2020. However, we have one more disease-specific symposium coming up on November 14th. And then our final Engage webinar is coming up on December 3rd, Caring for the Caregiver. So I would urge you, if either of those are of interest to you, to go to mda.org, and you could go to the Family Services tab and register under Engage events and to participate in those. We would not be able to put on educational programming without the support of our generous sponsors. So I would like to thank New Motion Foundation, Biogen, and Genentech for supporting this seminar. And then I have a few housekeeping items to go over before we begin our seminar today. We are recording the event, so we will be posting it to mda.org's website for on-demand viewing in a few weeks. And that will also ensure for those of you who have to leave during the presentation that we will be recording it and you can access that in a couple weeks. And then we are having a Q&A session at the end of each presentation. So please utilize the Q&A icon to type in your questions. If you hover over the bottom of your screen, a tray of webinar icons will appear, and just click on the Q&A icon. And you do not have to wait till the presentation is done. You can submit your questions as they come up, and then I will monitor that as the speakers are doing their presentation. So feel free to submit those whenever. And then we also encourage participants to communicate with each other by utilizing the chat feature if you'd like. If you just want to say hi or let us know where you're from, by all means, just make sure you click to all panelists and attendees. That way the attendees can see. And then we are going to be sending out a brief survey at the end. We are interested in your opinions, and we want to receive your feedback on what you heard today. And that will also ensure that for 2021 that we have all of your information on what you thought of the program so we can make adjustments in the future. So thank you in advance for taking that time to do that. Now, I'm going to introduce Dr. Manuela Cordy. She is currently an assistant professor and director of translational research in the Child Health Research Institute and Powell, excuse me, Gene Therapy Center at the University of Florida. Dr. Cordy is a clinical scientist engaged in translational research, focusing on understanding the contribution of neurological impairment in neuromuscular disorders by combining expertise in clinical assessment with novel therapies that rely on correcting the fundamental genetic defect. Her research interests also include outcome measures and clinical trial readiness for neuromuscular diseases such as Friedreich's ataxia, Pompe, and Duchenne. So thank you, Dr. Cordy, for being here this morning. I will go ahead and turn it over to you.
Hi, everyone. I am going to share my screen with my presentation. Here we go. And we can start from there. Do you see my screen?
Yes, we do.
Awesome. So thank you for the presentation. As it was mentioned, I work at University of Florida. I am going to discuss the way that I organize this presentation is with practical questions that usually the patient community ask relating specifically to gene therapy. Hopefully I will be able to answer some questions during the presentation, but at the end, there will be time to answer more questions that maybe I didn't address. So let's start with what actually is gene therapy. So gene therapy is a new treatment approach that has been shown in different clinical trials to be successful for different neuromuscular diseases. In order to understand what is gene therapy, let's start. Oops, my screen is not changing slide. Here we go. So let's discuss how we inherit information from our parents and what is the basis of genetic diseases. So the gene is that functional structure, that part of the DNA that code for specific protein. And the gene are created by different hundred and thousand of base pairs that code for a specific protein. The protein are made of lots of amino acid. So you can understand that a mistake, an error in the gene may lead to genetic conditions. So here is a video showing this concept. This is a functional gene that code for a functional protein. But when there is a mutated gene, like here in red, we might have an abnormal protein and therefore a condition that with symptoms. In some cases, it's more severe because the mutated gene is not producing any protein. In that case, we have a more severe manifestation. So the aim of gene therapy is to introduce a functional gene that will be able to make a functional protein that is, as I said, is function and it doesn't lead to any symptoms. So what do we use to bring this functional gene inside the body? We use vectors, and this is specifically in this presentation I will discuss of AAV vectors. AAV stands for adeno-associated virus. You don't have to remember this word, but just remember AAV is a virus that has specific characteristic and that's why this was chosen to bring this gene inside our body. First of all, it does not cause any disease. It's not pathogenic, so that's why we can use it. There are different serotypes depending on which tissue we want to target. So depending on the condition or the disease, we will choose one over the other one. And very important information about this virus is that it does not integrate in the DNA on some cell in the cell. Therefore, does not cause additional diseases. And also, the risk benefits is in favor of benefits, and that is being tested in several clinical trials. So this is the virus. The outside part is called capsid. Inside there is the genome. The genome is made of a functional gene that is depending on the condition we want to treat, and a promoter that control expression of the gene in different tissue. And the outside there is this part that is related to packaging of the virus. So when we inject the virus in the body, with different route of administration depending on the disease, the vector reach the cell encapsulate the virus, and then the virus reach the nucleus. The outside part of the virus stay outside here, and inside is released the genome, and the gene can produce a functional protein. So that is basically how it works. So just to go over against this concept, the gene therapy is a technique to correct defective gene that are causing different genetic condition. Introduce a new gene in the body, and we use a virus that does not cause any condition. So this is the different step that we went over on the video. So the virus is this one. It contains the gene of interest for that condition, is encapsulated inside the cell, and then it enters the nucleus, and inside the nucleus, the gene produces the functional protein of interest for that condition. So there are different types of gene therapy. The one that I just described is called gene replacement. So basically, we introduce a gene, a functional gene, that stay alongside to the mutated gene and produce a functional protein. But there are also others approaches that I'm not going to discuss in this presentation. One is gene addition. So basically, we introduce a new gene that alleviate some of the symptoms caused by the disease. There is gene inhibition. So we introduce a gene that inhibit another mutated gene that is overproducing the protein of interest. So this gene, in this case, inhibit the product of another gene. And then there is gene editing. In this case, we introduce a gene that correct the mutated gene that cause the condition. So in this presentation, I will really focus on the most used in clinical trial approach, and that is gene replacement. So we introduce a gene that stays alongside the mutated gene. So our AAV vector mates, here it is. This is a virus. The virus is, the genome is made by this red part on the side that is called inverted terminal repeat. You don't have to remember this term, but basically this stays for, this help the packaging of the virus. And then there are two other proteins, the rep and the cap. These are for replication and the stability of the capsid of the structure of the virus. So these parts are not really needed for our gene therapy treatment. So what we do when we produce this virus, we cut this part out, and this is substituted by something that we really need, is the therapeutic gene, so of interest. So depending on the condition that we are trying to treat, we put a functional gene and a promoter. The promoter is that part that controls the expression of the gene in different tissues. So if we want to restrict the expression of the gene in specific tissue, we will use a tissue specific promoter, so that control the expression of the gene in some organs and not in others. I just want to, like I have just few slides saying that this is really an exciting moment for gene therapy. This slide is coming for Peter Marks from FDA, showing that the number of IND, so the application for gene therapy to the FDA, is really increasing. So this one is 2017, 2018, and he reported that in August 2020, there were more than 1,000 active applications for gene therapy to the FDA. So it really is an exciting moment for gene therapy. And this correspond or predict more than 40 to 60 products that will be launched by 2030. And so there will be more than 500,000 patients that could be receive this treatment. So a very exciting moment for gene therapy. And in this slide, I want to show how the industry is really paying attention to the progress that we are doing in gene therapy. These are the trial, and the other part, like in the outside part of the circle, are phase 1, phase 2. That is the first trial that the FDA that we use to test a gene therapy product. You see there are lots of companies working on gene therapy, but just few reach marketing approval. And we can discuss, I have another slide here showing that a very exciting, there was in 2019 a very important approval for spinal muscular atrophy, SMA. This drug, this gene therapy is called Zolgensma. It's sponsored by Novartis. And so after this approval, lots of these patients with this devastating condition can receive this treatment. The one aspect that need to be addressed still in gene therapy, I'm not going to address a lot in this presentation for sake of time, but is the cost. As you can see, is very costly, and some of the insurance partners are really struggling covering this cost. And I'm going to address in the next slide why this cost is so high, and is every aspect is very expensive, but production of the gene therapy is the most expensive part of this process. So, and what are the challenges in gene therapy? I kind of already mentioned. The first one is the manufacturing, the production of the gene therapy is very costly, but there are also limitations that I'm going to address in the next slide. And the other aspect that is very important for the patient community is to understand that, that we might have some immune response to this virus that we use for gene therapy. So let's address the first challenge that we identify is the manufacturing. So the problem is really scaling up method that we use to produce the vector from clinical trial to commercial size. When a product become commercial, the number of individuals receiving is, of course, increasing. So we really need to improve the production. Currently, the production of this vector, of this gene therapy, is based on cell line. Basically, we use cell to produce and multiply this virus inside a cell. So you can understand that we need a lot of cell in order to produce a lot of virus. So this process currently is still inefficient to scale up to a commercial use of this gene therapy. And this process, you can identify, is very costly because we need to repeat several times this process of producing the vector within the cell. So the FDA is actually working on this aspect and is collaborating with different institutions and different companies to develop a more streamlined way to facilitate manufacturing, to reduce the cost and make sure that there will be a vector produced for all condition and all all condition, even those conditions that don't have a very large marketing size. So for those conditions that are ultra orphan, for example. So this is another slide showing the problem of manufacturing. So in clinical trial, we produce, this is an example of the Starbucks size. So we have a tall, a grande, and a venti cup here. So currently, we can, the current platform can manufacture for a tall kind of size, so between 1 and 100 patients. But what we want to really scale up these methods to be able to treat more like 10,000 and more patients. So we really want to scale up our the manufacturing to technology that is easier and reduce the cost and is easier to provide this treatment for as many patients as we want. So this is one of the challenges that bring the cost of the drug high and is currently being addressed by several investigators and by the FDA. The other challenge in gene therapy are, as I mentioned before, is the immunoresponse to AAV. So there are three topics that I want to address in this next slide. First, and they are connected to each other. The first one is the antibody formation against the AAV and the transgene. The second is the pre-existing immunity, or some people call it pre-immunity to AAV, and repeat dosing. Just to throw a joke there, why don't ants get sick? Because they have little antibodies. So this slide introduced the topic of antibodies. So when we have, introduce a foreign foreign agent inside our body, our immune system produce this protein called antibody that have this white shape, or they're also called immunoglobulin. And these proteins are made to attack intruders in our body, which are called antigen. So each antigen has a specific antibody that latch onto it and neutralize the antigen. So if you think that this antigen can be our AAV that we are using to introduce the gene inside the body, you can understand why these antibodies are not that good in gene therapy, because it will neutralize the treatment. And so when there is this reaction, which is called immunoresponse, there are also some symptoms, like for example, in fever, nausea, and others symptoms. But to try to understand better how this affects gene therapy, so I told you that when we introduce the AAV in the body, there is a primary immunoresponse with an increase of antibody. But then if this is a second exposure, so if this is a second time that the body is exposed to this AAV, this reaction of antibody is exaggerated, as you can see from this red line. And this is a big problem in gene therapy. Because all these symptoms that I mentioned are exaggerated. So there is a more severe immune response, and also the treatment is neutralized, so it's not really working. It's not the efficacy of the treatment is really reduced. So why this is important to explain that if there is a second exposure, because AAV is a virus that is present in the environment. So lots of people are already been exposed to this virus, especially we did a study, and other people did a study. We identified that about 60% of the individual are already exposed to this virus. So in those patients, the treatment might be not as effective, and it might also be cause some severe immune responses. That is why lots of clinical trials use a threshold, a threshold of antibody level that as an exclusion criteria. So even if an individual has a pre-existing immunity to AAV, so was already exposed to AAV, they are most likely excluded from gene therapy trial. And this is really limiting the number of subjects can benefit from this treatment. So our group at University of Florida and others in other universities are really working to try to understand what is this threshold that is being used in clinical trial, to understand if this can be adjusted and to include more individual to this treatment. So if you look in this graph on the right here, subject C is that subject that is lucky because hasn't been exposed to AAV yet. So his antibody titer is low, is below the threshold, so he can receive the gene therapy. But we might have a subject B, that is just above that threshold. So current clinical trial will exclude this subject for receiving the gene therapy. So we are also working on identify an immunomodulation regimen to reduce this titer below a threshold so that it can receive the gene therapy. So these are ongoing study that we show in preclinical work that we can definitely reduce this antibody titer below the threshold. And then we are also working on identify what is the maximal threshold, maximal level of antibody that can be reduced. For example, this subject A that is unlucky because it was already exposed to AAV, has a very high level of antibody and might not be able to be included in any gene therapy, even after treatment of immunomodulation. So is gene therapy durable for the lifespan? Well, maybe, or probably not. So this depends on the condition. Because there is lots of regeneration in our body. For example, the muscle goes through lots of regeneration. So the vector, when we with those individual, is diluted throughout this regeneration. This is not true for CNS diseases because there is not lots of regeneration in CNS. So when we most likely it will be durable for CNS condition and less durable for muscle condition or liver condition where there is lots of change within the tissue. So this brings up another issue. So early exposure is probably less durable. So and this is important with newborn screening. So the diagnosis are done earlier. There will be an early treatment, but this leads also to less durable treatment. So to convince you that this is not durable, we did an experiment in primates. We dosed primates at birth with a gene therapy. And then we did muscle biopsy every month for a year to test the level of the transgene activity in the muscle. As you can see, reduce all over the first year. So really, and this is due to growth of the animal. And this is what we think would happen in a growing child that received the treatment at early time points. And again, this is more relevant to muscle and liver condition and less relevant to CNS the pathology of the nervous system. So just to recap a little bit of this concept, so when we introduce a foreign agent inside the body, there is an immune response against this vector that we use, and this primary antibody for response affect the clearance of the vector because it's neutralized, and therefore affect the efficacy of the treatment. But also is important because there is this antibody response, it precludes the repeat dosing of the same gene therapy. I showed that slide where the second exposure of the vector increase enormously the immune response. So this precludes the repeat of gene therapy. But the good news is that our group is studying a way to overcome these challenges. So we tested an immunomodulation regimen that is given before the AAV exposure, before the gene therapy. And basically this regimen block the, this immunomodulation block the antibody response, and therefore we prevent all those processes that I showed you before. And I don't want to go into detail of the immunomodulation, but what we are blocking those cells that produce antibody. And it's important to also understand this is not a permanent block, it's just a transient block just at the time of the gene therapy. And these drugs are safe, and they are also used for other autoimmune diseases chronically. In this case, will be just one time for the gene therapy. And so using this immunomodulation, we could also allow for repeat dosing of gene therapy to reach a target high dose. And so we ran this study. This is in humans, specifically in Pompe disease. You can see this black solid line here. These are 6 patients that received the gene therapy without immunomodulation, and they have an increase of antibody against the that gene therapy that we use. In that case was AV1. But there are a subgroup of these patients that had were receiving immunomodulation for different reasons, and then you can see that the antibodies stay low. So we really were able to block this immune response. So we perform lots of clinical study, and we submit a package to the FDA asking for approval to for the first time in history to dose the gene therapy twice in the same individual, using this immunomodulation to block the antibody. And so this is an ongoing study in Pompe disease. Is an adult patient where we dose one leg and with the active treatment and the other leg with a placebo. And then 4 months later, the leg that received the active treatment received the placebo, the other leg received the active treatment. So basically in this study, one individual would receive the gene therapy twice with the immunomodulation, with the purpose to prevent the antibody formation. And so in this slide, I'm showing you we were successful. These are the antibody in this patient that received the gene therapy twice. They remain low compared to that other patient that received just one dose of gene therapy without immunomodulation. So we were able to successfully dose the gene therapy twice with the help of the immunomodulation. Just to go over why this is relevant and what is the messages I want to deliver is that managing of the antibody response in gene therapy is very important for both the primary response when we introduce the vector for the first time, but also when is the second time. So this is important relating to pre-immunity. Some patients are already exposed to AAV and they will be excluded from gene therapy trial, but if we use the immunomodulation to bring that antibody below that threshold, they will be able to participate to studies. This is very important for early intervention and relating to newborn screening. And this also lead to incremental dosing to reach a very high dose that could be toxic if we introduce it all together, but it might be less toxic if it's divided in multiple dosing. So what are the side effects of gene therapy? So as I mentioned before, we can have fever, nausea, Also, our blood count will change. We will have also a decrease of platelet, an increase of liver enzyme with the liver inflammation, and a generalized immune response. There might be also some late effects that are not known. So far, gene therapy have been shown to be safe in clinical trials. So take-home messages for this presentation. So gene replacement is a successful and safe treatment strategy. The production of AAV is very costly, and we are working, the FDA and other groups, also our group is working on identify a platform to scale up this production to really a large scale for commercial use. The immunomodulation is successful to block the antibody that attack the gene therapy, and therefore this increase safety and increase also the efficacy of the treatment. For some diseases, gene therapy is not durable, and so if we use immunomodulation, we are able to allow repeat dosing, especially when the dose is done early at early age. Some individuals are less lucky because they are already exposed to AAV, but we have a way to manage this pre-immunity with an immunomodulation before the exposure to the gene therapy to reduce this antibody titer below a threshold so that they will be able to enroll to gene therapy trial. And there are some side effects and risk with gene therapy, but these side effects are really are very minimal compared to the beneficial of the treatment that can provide. So this is my last slide, and I want to show you why the development of gene therapy might take years and why it is costly. So the way it works, we when we identify for those conditions that we identify the gene that cause the disease, we perform some proof of concept study in the lab, introducing this gene inside an animal model, usually is a mouse. When we see that there are an improvement of the condition, and we assemble what is called an IND, which is an investigation on new drug application for the FDA. So this IND include a clinical protocol, the plan that we want to do in humans, a manufacturing plan, so how we plan to manufacture the drug with lots of specific on testing to make sure that the drug is safe, and also a very detailed plan of all the preclinical study that we want to do. Usually we use two model, a small model like a mouse or a rat, and a larger model like a primate or a dog, to test the safety, the toxicology, and the biodistribution of the vector. So we submit all this plan to the FDA. This is called pre-IND package, and the FDA set up a meeting with the team that is working on this project to discuss some issue relating to the package that we submitted, and then then you go ahead and perform all the preclinical study that were described in the pre-IND package, based on the feedback that we receive from the FDA. And when we have all these results, we put together this final IND package and we submit to the FDA. And the FDA review everything and give the okay to proceed with a phase 1 to a clinical study. So the most costly part, everything is very costly, especially working the primates, but the most costly part is really the manufacturing of the drug, as I mentioned before, that really bring the cost of the drug super high. And the FDA and lots of scientists are working to identify a platform that reduce the cost and is more efficient to produce more gene therapy for more individuals. So as I said, the plan that we always have is look like a straight line to the finish line, but in reality, there are lots of challenges that we run into conducting this experiment. But as I mentioned early, is a very good era for gene therapy since there is lots of progress. So this is our team at University of Florida working on all these gene therapy projects. This team is led by Dr. Barry Byrne, and especially thanks to all patients that really participate to this clinical trial, sometimes not knowing even the side effects that are related to this treatment. So big thanks to all of you for participating to this study, but also to listen to this presentation.
Thank you, Dr. Cordy. You know, I was just curious, the gene therapy that you just talked about, is that general timeline, is that accurate for all types of muscular dystrophies for gene therapy, or is that targeting a certain type of muscular dystrophy?
That's an interesting question. So there are different answers to this question. So when the gene is known of causing the disease, usually this pathway is easier, because the gene is known, as I mentioned before, you produce this preclinical test and then you get to clinic. But then there are also different issues relating to this aspect, relating to being this condition ultra orphan. Sometimes the experiments are done in the lab and are successful, but the next step of launching those treatment to marketing approval or to commercialization is a different, because usually that step is done by a company. This is a very interesting period for gene therapy also because the FDA is really focusing on those ultra condition, so unspoken condition, and they are really trying to identify a platform where those conditions that are ultra orphan can be treated with taking all those information coming from other conditions that are maybe a little bit have a bigger size, and so we are learning for other conditions to apply also for those ultra orphan. I don't know if that's answer your question. And then there are different other conditions where the gene is not known, or the editing is a little bit more complicated, and then we can go in the specific of the condition, but that is usually the general approach.
I think it's fascinating that we are able to even do this. It's just such an amazing therapy. It's really amazing. I don't see any questions that have come through, so thank you very much for your time this morning on Saturday.
Thank you. Thank you everyone for listening.
Have a good rest of your day.
Thank you.